You can see group 1, Brittany, Megan, Sienna. Group 2, Austin, Karina, and Taylor. And Taylor is the woman who came on testing, but left off. Group 3, Janessa, Layla, and Makai. And I let Canvas do this randomly, okay? David Streeter, again, I haven't seen him. I tried to email him and no one seems, I don't know if you guys know him, but if you do, please have him email me. But John and Julie, and then Deanna, Mariana, and Milana. Carla, Isley, and Jasmine. Christian, Jacob, and Jamie. Colin, Jared, and Lily. I was like, how did you three end up together? Imagine. Adrian, Natalie, and Sarah. Okay, so you guys can, those are your groups. I don't know if you guys want to, you don't have to study together, you don't have to do anything with your groups, but that's who you'll be working with on your exam. If someone's not here that day, then remember, I'm not going to be here on Wednesday, but we'll have it get worked out. Okay. Okay, questions? Okay, so last week we beta oxidation, right? Oxidation of fats. And we looked at how we break those down. How did it go for people? Fatty acid metabolism, beta oxidation. Any questions come up for you guys? The kind of weird thing was like we can't make glucose from fat, right? That's kind of the big thing. All right, we are going to move on. I'll have office hours tomorrow on Zoom from 7 to 8. I think I might have a second office hours tomorrow from 1 to 2 in person, if anyone wants to, because my other classes also have an exam on Wednesday. And so maybe there'll be a lot of people in office hours. Yeah, Austin.
On the second practice exam, there's a lot, like a few questions on like glycogen and insulin. Are those going to be tested on this exams?
Yeah, so it's hard for me to separate them. So yeah, so definitely something that was different about this semester is that glycogen, usually like we haven't covered it for the first exam, but I kind of moved things so that it was all together on the first exam. So there might not be as much. But yeah, always be ready for, like, I might still ask you about glycolysis. I might still ask you about gluconeogenesis. They're all connected, especially this fatty acid metabolism, right? We saw how you have the bioxalate cycle, succinate with the citric acid cycle, with swap oxalate to make glucose in gluconeogenesis. So they're all connected. Yes, the focus will be citric acid cycle, beta oxidation, but I definitely like to think about things in a big picture and how they're connected. Other questions? Okay. I'm going to start talking about amino acid oxidation. And when I remember first looking at this topic and thinking like, oh gosh, the beginning of this section was so confusing. And so I made this like skeletal outline notes. It's on the Canvas page. So if you want to do this like on your iPad and do it digitally, then you can fill this in digitally. So I basically made like kind of an outline of my notes and then left some blank spaces so you guys can follow along and fill in as we go. Might be and like I said, it's online as well, if you'd rather feel that. It just kind of to help us. Sometimes people are like, you do that for every class. Put a 2-page. Okay. Anyone have interest? Okay. Ooh, I think I have a. All right, so. We're moving into amino acid oxidation. And so now we're gonna be looking at urea being produced. Okay, so some percentage of our energy does come from amino acids. Okay, and remember, amino acids are like the building blocks of proteins, right? You take a bunch of amino acids, you polymerize them, and you get a protein. And then remember, some proteins are enzymes. Okay, sometimes I feel like that connection can get lost sometimes. Again, depending on the organism. So if like you're a carnivore, like you're a lion, and like 90% of your energy is going to come from amino acids. Herbivores, a smaller fraction. Okay. Some microorganisms might scavenge amino acids. They're very little. In plants, it's very rare to have amino acids be a source of energy in the plants. Okay. All right. Here we go. So there's some circumstances when animals are going to undergo amino acid oxidation. So we're starting to look at another catabolic pathway here. Okay. So we already looked, remember the three main sources fuels are carbs, fats, and proteins. Okay. We looked at catabolic process of carbs, that was glycolysis, right? We just finished catabolic process of fats, that was beta oxidation. Now we're starting to look at the catabolic process of proteins, more specifically amino acids. Okay. When are animals going to do this amino acid oxidation? Okay, kind of if there's some leftover amino acids from normal protein turnover, okay. So there's normal synthesis and degradation of cellular proteins. You can recycle them, okay, if you need to. If there's a diet rich in protein, okay, then we're gonna do some amino acid oxidation. So again, if they're taking in more amino acids than the body needs for protein synthesis, okay. And then during starvation and uncontrolled diabetes, okay. Remember, uncontrolled diabetes, the body does think it's starving, even though you might be eating a bunch, it cannot, the cells are not recognizing all the energy or like glucose in the blood, okay. So proteins in the body can be broken down to supply amino acids for energy, okay. So if carbohydrates are scarce. All right. All right, so you guys should be following along and filling in, right? Okay. And so the first one, sorry, I missed the first one. So we're going to be discussing amino acid oxidation in vertebrates. Okay.
Sorry, right. Invertebrates or invertebrates?
Vertebrates. The okay, and then this answers that next question is when do amino acids get oxidized? This is when they get oxidized. Okay, so normal synthesis and degradation, a diet rich in protein, and under starvation and uncontrolled diabetes. Okay, so now there's one distinguishing feature. If you guys remember, do you guys remember the general structure of an amino acid? Hopefully from some other biologists, you've seen these, okay? So like glutamate's an amino acid. Remember, they all have this alpha carbon in the middle, and then they're all attached to a hydrogen, and then this amino group, and then a carboxylic or carboxylate group, okay? And then there's a R group, right? This part can vary depending on which amino acid you're looking at, right? Here's, okay, here, like right here, this is like our general form for an amino acid, right? You guys remember this? You guys seen this somewhere else? Yeah, okay. Okay, so there's kind of one distinguishing feature there that on these amino groups that we haven't seen in our fats, and we didn't see it in our carbohydrates. Like, what do you guys notice? What's different about it? It's charged. Okay. What parts are charged?
The difference of the.
The nitrogen. Okay. The nitrogen. We haven't seen anything with nitrogen in it yet, right? Fats are carbon, hydrogen, oxygen. Glucose, carbon, hydrogen, oxygen. Okay. So we got this nitrogen there that we got to handle. Okay, so that's kind of a distinguishing feature here with our amino acid oxidation. We have this amino group, and we have to separate it from the carbon skeleton. Okay, we have to separate from the carbon skeleton. So, in metabolism, we can first deal with that amino group. We can either recycle it and use it, or we can excrete it. And that's what the urea cycle is for. Okay, so the urea cycle is a little different than like citric acid cycle, right? It is a way to get rid of excess nitrogen. And we'll go through that. Okay. And then what we have left over is this carbon skeleton that we can break up and use. Okay. All right. So again, the key difference is that amino group. All right. So this is an overview of what happens of amino acid catabolism. Again, the breakdown of amino acids. So you have intracellular proteins, right? You have proteins in your diet, and they can break down to amino acids. Know that amino acids can be, this is a double-headed arrow, right? It can go back and be used to make more protein. Okay. Again, we have to remove the amino group. Okay, that's what we just said. We have to remove the amino group. And then we have this carbon skeleton. Okay. The amino group can be used to make more amino acids or nucleotides to other molecules that need nitrogen. Okay. Or they can be incorporated into a molecule we call carbonyl phosphate, which we'll see is part of the urea cycle. And again, that goes into the urea cycle, and we excrete excess nitrogen as urea. Okay. The carbon skeleton, that what's left over, we call alpha keto acids, and most of them can feed into the citric acid cycle. Okay, and get fully oxidized to CO2 and water and ATP, or be used, right, to get to oxaloacetate and go through gluconeogenesis to make glucose. And these two, the citric acid cycle and the urea cycle, are connected through what we call this aspartate argininosuccinate shunt. Okay. And we'll see what that means once we look at the urea cycle. We'll try to see the connection between the urea cycle and citric acid cycle. Okay. All right. So I don't have a. Keep thinking I can click. Not click. Okay. So the first step. So I haven't started talking about the next part, the breaking down of dietary protein. Sorry. Okay. So the first step of degradation for all amino acids is, again, we're going to have to remove that amino group. Okay. And there's a couple important amino acids that play key roles in this process that you should be aware of. Okay. Glutamate. Okay. We'll see glutamate a lot in here. Glutamine is another important amino acid we'll see a lot. Alanine. Okay. Is going to play an important role here. Okay. And aspartate. Aspartate. Okay. So glutamate and glutamine are present in higher concentrations, okay? Because you're gonna see that they play a key role in helping transport our amino groups, okay? All right. You guys can go through this more thoroughly, but again, we're gonna be talking about vertebrates. And so depending on what type of organism though, that excess nitrogen might be excreted differently, okay? And so some, it might be excreted as ammonium, uric acid, so this is birds and reptiles. And then for us vertebrates, we excrete that excess nitrogen as urea. That's how that excess nitrogen is removed from our body. Okay. Now, okay, so the words are on the previous slide if you want to look at it. I like looking at the images though. So let's think about again those sources of the proteins. Okay, so the first example here is our dietary protein, right? So you eat protein, some form, let's just say you're eating, I don't know, a steak or something. You chomp it with your teeth, right? Break it up. Okay. With the starches, with like carbohydrates, you have enzymes in your saliva that start breaking those up. But that's not happening with proteins. Okay, it's not until it reaches the stomach. And you have hydrochloric acid in your stomach, okay? And it's very acidic, like pH 2 in your stomach with the hydrochloric acid. And we know that acidic environments can denature your proteins, okay? So that low pH in the stomach will start to denature the protein. Okay? Now, when that protein starts to denature, do you guys remember what's connecting each amino acid to make up a protein? You have these peptide bonds, right, between the amino group of one and the carboxy group of another. Make that amide bond or a peptide bond that connects one amino acid to the next. So no, as that protein begins to denature in this acidic environment, those peptide bonds become more susceptible, okay? And they're more accessible to hydrolysis, okay? In the stomach, we have pepsinogen that gets converted to pepsin, okay? And so pepsinogen is what we call a zymogen. So let's see, let me make sure I'm following along. So makes protein more susceptible to hydrolysis by pepsin, okay? That's in your little worksheet. And we have this inactive precursor, you can see it on your notes, it says zymogen, right? And the zymogen of pepsin is the pepsinogen. So it's this inactive precursor. Because again, this is something that's going to eat away at proteins. Remember, we've seen all the enzymes that are so important in our metabolic pathways. We wouldn't just want pepsin acting on all these important proteins in our body, okay? It has to be active at a certain point. So pepsinogen undergoes this autocatalytic cleavage, okay? So it cleaves itself to form the active form pepsin. And pepsin is going to hydrolyze peptide bonds, okay? It's a protease. So it's going to hydrolyze peptide bonds on the end side of aromatic amino acids. Okay. So again, pepsinogen in the stomach gets converted to pepsin. And again, which is active and hydrolyzes the enzyme of aromatic amino acids. So this is going to create smaller peptides. Okay, so we're breaking up our protein, hydrolyzing some of those peptide bonds that create smaller peptides. Those peptides are going to continue on, okay, into the small intestine here. Okay. Bicarbonate is going to get released in the small intestine, and that's going to help neutralize the HCl. So I always think it's interesting, like our buffer system in our body, right, is uses bicarbonate, okay? And then there are even more proteases released in the small intestine, okay, that can further break down this protein. So there's several proteases that are released. And again, they get converted from their inactive form, from the zymogen, to the active form, the actual active protease that can go and continue cleaving more peptide bonds.
And
Yeah.
Is that the hormonal signals secretion is identified?
Are you still on the first page?
Yes.
Oh, yeah. So hormonal signals, secretion of zymogens by the pancreas. So the pancreas is the one releasing the zymogens, okay, into the small intestine. There's this pancreatic duct that these zymogens are releasing to small intestine through. Okay. Yeah.
I was wondering, is pepsinogen a type of zymogen?
Yes. So there's trypsin. You might have heard of trypsin before. So trypsinogen would be the inactive precursor, the zymogen of trypsin, right? That would be more in the small intestine. And so pepsinogen is the inactive form of pepsin. But yes, they're all zymogens, like the general term, and then pepsinogen like specific. It's the general term for these inactive precursors. So zymogen is an inactive precursor, so it's like a protease that's not active yet. So zymogen just means inactive protease. Other questions? Yeah, Carla.
Wait, my hands, so.
What is that? Into the small intestine. Yeah, so again, the pancreas is secreting the zymogens through this pancreatic duct into the small intestine. Yeah.
What is the key difference from other food sources?
The amino group. Yeah, the amino group. The nitrogen. Okay.
It says the inside of aromatic proteins.
The inside of aromatic amino acids.
Okay, is it specific for aromatic?
Yes. Pepsinogen is.
Okay.
Yeah, pepsinogen is pepsin is active. So remember, different proteases. I think in 445 you might have covered that. Biometers, I don't know if you guys covered that other places, but there's different proteases that have different specificities. They'll recognize different amino acids and cut after certain ones. There's a whole table of them, like in Leninger, there's a table in an earlier chapter that will say, like, here's a protease, this is what it's specific for. So pepsin is specific for the inside of the amino acids. I can't tell you what the other ones are specific for. I think like it's like that's a fun that sometimes people do that in classes, like, hey, we cleaved with this, we got these sequences, now figure out what the original sequence was. I don't know if you guys did that, like in 445, but
445 structural.
yeah, that's first semester biochem. It's like the first half of the text, of the line your text. All right, other questions? Sabrina?
Did we do the, now we have a mixture of three?
Haven't done that yet. Okay, so right, so we've been cutting up the protein as it travels through the stomach, as it travels through the small intestine. It's been susceptible to these proteases that are cleaving the peptide bonds. Again, that's the amide bond between one amino acid and another amino acid. Okay, so yes, now we have a mixture of free amino acids. Okay. And then they can enter the blood through the lining of the small intestine. Okay, this intestinal mucosa. and absorb amino acids. So we now have a mixture of free amino acids that can enter the blood, the lining of the small intestines, and from there, they can travel through the blood to the liver. They can travel to the liver. Okay, so yeah, you can hear people flipping. So how does anyone have a question about that first page of kind of filling in the skeleton of? Yeah. Uh-huh.
And then same.
Yeah, yes, exactly. Okay. Okay, so. We're gonna see more about this later, but pyridoxal phosphate helps in transferring that amino group. Remember, we said we gotta handle that amino group, so this molecule pyridoxal phosphate participates in the transfer, okay? Sabrina, I think it's your 441 project that requires pyridoxal phosphate. I think it's yours, and it's not yours, Natalie. Let's see who's in there, maybe. Is it yours too? Dr. Kudo keeps asking one of your groups, like, who needs pyridoxal phosphate? Like, why is it there? Well, this is where it comes up in biochem. And so Julia is your partner, Sabrina, and she was in this class last year. But I feel like she doesn't remember this part. But pyridoxal phosphate carries amino groups, okay, and we're gonna see that in a bit. Okay. All right. So we just went through this digestion of this protein that we ate in our diet. Okay, we have now crossed into the blood through the small intestines. Okay, and traveled to the liver. Notice we're in the liver here. This looks like the liver. Yeah. Okay, so notice we are in the liver, and we're doing the first step in catabolism of most amino acids. Okay, so the first step of catabolism for amino acids is to remove the amino group. Okay, and again, the molecule that's going to help us do this is that pyridoxal phosphate, sometimes called PLP. Okay. Okay. And so the enzyme that is going to do this is aminotransferases. Hold on, let me. So we're going to remove, so we have some coming amino acid. We're going to remove the amino group, okay? The enzymes that catalyze this reaction are called aminotransferases. And again, the required cofactor is PLP. Okay, that's that pyridoxal phosphate. So what's going to happen here? So this alpha amino group, okay, is going to get transferred to alpha ketoglutarate. Okay. This is alpha ketoglutarate. Ever heard of alpha ketoglutarate before? Yeah. Okay. Remember alpha ketoglutarate, we saw that in the citric acid cycle. Okay. Alpha ketoglutarate picks up the amino acid and picks up the amino group and becomes L-glutamate. Okay. Whatever amino acid you have coming in from your intake of dietary protein loses the amino group, and you're left with the carbon skeleton known as the alpha keto acid. Okay. So on the second page, we're removing the alpha amino group. Okay. Enzymes that do this are called aminotransferases. They're also called transaminases. Okay, so you might see that term as well, transaminase. Okay. And then in the box, right, the top box is actually supposed to be the alpha keto acid. So this might be opposite of what you see on your worksheet. Okay, the alpha keto acid here. And think of that word, alpha keto acid, right? It's carboxylic acid, you have, it's a ketone, okay? Alpha keto acid. And then on your worksheet, you have alpha ketoglutarate on the bottom right, okay? And it is getting transformed into glutamate. So this is the structure that goes in the box on the bottom right here. Should be alpha, should be glutamate, sorry, glutamate. Yes, and what are the requirements? Those circles above and below, okay? Remember, you need, the enzyme is aminotransferase, and then the cofactor is PLP. Okay. So why are we doing this? Okay. Again, we've taken in this source of amino acids. We're at the liver. We're starting oxidation. First thing we're doing is removing the amino group. We're transferring it, okay, to alpha ketoglutarate. So why? To collect the amino groups to the same amino acid, and then that amino acid can enter the urea cycle, or it can be used in biosynthesis. So again, this allows for the collection of amino groups from many different amino acids to just glutamate. So it's just one amino acid that feeds in, okay? So glutamate serves as an amino group donor for synthesis and for excretion. So instead of having 20 pathways for excretion and 20 pathways for biosynthesis, right, it can all just funnel through glutamate.
You said it was an intermediate donor for.
It's a donor for synthesis and excretion. So these aminotransferases, okay, they tend to be specific for alpha ketoglutarate as the amino acceptor, okay? But they can differ. There's different aminotransferases depending on which amino acid is donating the amino group here, okay? And they're named after that, okay? So you might get like an alanine aminotransferase, okay? Good question?
Okay, I think I just need to restate it to make sure I understand.
Okay.
So there are many different aminotransferases, and they can all take different types of amino acids and convert them to L-glutamate that only have one exclusion pathway instead of another.
Yes. Yeah. Okay. And if we go back, so before this, I had a slide of the urea cycle, okay? So you could kind of see what's happening, and what you see is glutamate. So this is amino acids coming from all kinds of different places, and we'll look back at that, okay? But glutamate feeds in, this is the start of the urea cycle. It needs to make carbonyl phosphate, and then it'll feed into the cycle. But again, it all starts here at glutamate. So all our different ways of getting amino groups, acquiring amino groups. Again, if we can get the amino groups of glutamate, then that can feed into urea cycle for excretion. And right now, what we're like this slide that we're looking at, I think I'm gonna, this one is showing you this part of the, yeah. This part, right, we're seeing amino acids transfer their amino group to alpha ketoglutarate. That makes glutamate, and from the amino acid, we get the alpha keto acid, right? So we can like see this little piece right here. That's what this is showing. Okay. So again, depending on which amino acid, the amino transferases can have different names, depending on the different amino acids, okay? Yeah, Sabrina?
So is it what belongs in these circles?
Yeah, so in the circles, right? Aminotransferase, that's the enzyme that catalyzes this reaction. And then PLP, we're gonna start talking about PLP. PLP is always the prosthetic group of aminotransferases, okay? So they always have pyridoxal phosphate, that PLP. Yes. Yes. It's always as a prosthetic group. It's an intermediate carrier of amino acids as opposed to, let's see. Let's look at it. Okay. So again, it's an intermediate enzyme-bound carrier of amino groups. Okay. It has an aldehyde group that can reversibly react with amino groups. Okay. Once it accepts an amino group, it becomes aminated before this pyridoxamine phosphate, and that can react with carbonyl groups. Okay. So. Pyridoxal phosphate is linked, okay, covalently linked to the enzyme. So here's our enzyme. It has a lysine residue with that amino group that can attach to the carbon of the carbonyl of the aldehyde, okay? And so you form this shift base. So again, the PLP is bonded to the enzyme through a shift base. Hey, this is more detailed than I asked in your notes, okay? But again, just so you guys are aware of how this molecule interacts with the enzyme. Okay. Okay. So again, it's bonded to the active side of the enzyme through the formation of the shift base. And so we get this interesting action happening here, okay? So here's our aminotransferase, okay? The whole enzyme, I feel like this is really misleading to students, right? This is a big enzyme made of many amino acids, right? Again, on the active site, you have a lysine residue with its amino group. It forms that shift base with the pyridoxal phosphate, okay? So this is all in the active side of the enzyme. You have your amino acid coming in to donate its amino group. We got to handle that amino group. This is how we handle it, okay? And so it can interact, okay, with the carbon here, okay? So it forms, notice that PLP was forming a shift base with the enzyme. Once the amino acid enters into the active site, now that PLP is forming a shift base with the amino group from the incoming amino acid. Okay. We get a side chain of one of the other amino acids acting as a base. It abstracts a proton. Okay. We see the electrons getting pushed. Okay. We form this quinoid intermediate. And then we go through this rearrangement. Okay, the important part here, the important part I want you guys to see is that what leaves is the alpha keto acid. It left behind the amino group on the PLP. Okay, It's like the alpha keto acid comes in and passes the amino group to PLP. It's like, hang on to this. Okay, and then it leaves the alpha keto leaves, the alpha keto acid leaves as the carbon skeleton. Okay. Then, so we're over here. Okay, alpha keto glutarate, which is an alpha keto acid, can enter into the active site and go through the reverse process here. Okay, alpha keto glutarate comes in, picks up that amino group through the reverse process, and it will leave as glutamate. So this process that we just saw, again, alpha keto, an amino acid, right? Donating its amino group to alpha keto glutarate. Okay, that's how it happens, again, through the PLP at the active site of that aminotransferase. Yeah,
In this schematic, I noticed that the ELP is down to the enzyme. and
Uh-huh.
The amino acid is free floating, but I think in the 2 slides back, I think the PLP is shown, the top structure is, because I think the
Yes.
PLP is shown without being bound to the enzyme, and I was wondering, are they both bound to the enzyme? They just,
what do you mean both, the amino acid and the PLP? No, so just the PLP. So this is showing you how the PLP binds to the enzyme. Once it's bound, right, it forms a shift base, so there's the PLP bound. Right, so that's just showing formation between the enzyme active site and the PLP. And that's where we're starting here. Right, the PLP bound through the shift base to the enzyme active site.
okay.
All right, and then the amino, so then again, whatever amino acid is gonna need to donate its amino group, comes into the active site. Do a series of rearrangements, ends up donating, right? So notice in going from here to here, now instead of PLP being bound to the enzyme, it's now bound to that incoming amino acid right here. The amino group of that amino acid more specifically. Okay. We get electron rearrangements. And then again, the carbon skeleton leaves as an alpha keto acid. And the amino group stayed behind, bound to PLP. Now to get the second part where you get alpha keto glutarate going to glutamate, you got to go from right to left. This figure's in your text, read the caption carefully. So you have this pyridine ring, okay, of PLP. This is a pyridine, okay? You have the nitrogen in there. That pyridine ring can act as an electron sink during this process. Notice we get a lot of delocalization of electrons in that pyridine ring, okay? And that's going to help stabilize these intermediate processes. Okay, so it's that pyridine ring of the PLP that can act as an electron sink and get electron delocalization of the electrons. This is called a ping pong reaction, okay? The first product reacts and must leave before the second substrate can come in and bind, okay? So again, the incoming amino acid binds to the active site, donates its amino group, okay? And then it departs as the alpha keto acid. And then an incoming alpha keto acid binds, accepts the amino group, and then it departs as an amino acid. Okay, so one comes in, leaves, another one comes in, picks up the amino group, and takes off. So it's like an amino acid comes in, like, hey, PLP, here's my amino group, right? Hold on to it. Someone's coming for it in a bit. It takes off. Then the alpha keto glutarate comes in. It's like, okay, yeah, thank you for holding that for me. I will now be aminated and leave as glutamate. Okay, let's look at your let's see your worksheet. So again, PLP is an intermediate carrier of amino groups. It's at the active site of aminotransferases. This is a ping pong reaction. Forms bond to the alpha amino group. Through the shift base, it stabilizes the carbanion. PLP is highly conjugated, okay? Acts as an electron sink, it can accept electrons into a conjugated system. Electrons rearrange, it leads to hydrolysis of a new shift base. It gives the alpha keto acid leaving the amino group on PLP. Okay. We've gone through page 2. Now we're on page 3 of the little handout I gave you, okay? Now another alpha keto acid can enter the active site. Again, usually that's alpha ketoglutarate. And then the reverse steps lead to formation of an amino acid. And that would be glutamine. Okay, in parentheses there. Okay. So that's the first step of oxidation for a lot of amino acids. Again, it's removal of that amino group. The way we remove that amino group is with aminotransferases. We transfer the amino group to alpha ketoglutarate. Okay. And again, the required cofactor is PLP there. Questions on that? But there's a lot of information on amino acid oxidation. Did you guys, again, if you read this section, I feel like it's a lot of information too. Okay, yeah, question.
I think I missed it, but did you ever explain why alpha ketoglutarate specifically is the amino?
The amino group of choice. You know, I think I'll talk about it later, but the idea alpha ketoglutarate, I think is actually shown on your next. So glutamate can go under further amination, and we'll talk about this more, to glutamine. And when it's converted to glutamine, then you're carrying two. You can pick up another amino group in the side chain of glutamine. So we said glutamate and glutamine. So I think again it allows for a further amination and a way to carry more amino groups to the liver. Efficiency now. Okay. Other questions on this? I know it's a little weird. But again, it's very interesting, right? It's like the active side. One comes in, hands off the amino group, it leaves. The other one comes in, picks up the amino group, it leaves. PLP, intermediate carrier of that amino group. Okay. So there's some other reactions. Again, I'm not expecting you to know these, but PLP also catalyzes racemization of amino acids. Okay, so we can go from the L amino acid to the D amino acid. Okay, so PLP can help carry that reaction out. It also does decarboxylation of amino acids. Okay, so here again, we have our incoming amino acid. It can go to this pathway C, and then you can have removal of that carboxy group. You can see it getting removed there. So different roles PLP can play. Okay. So now, glutamate has collected, right, all these amino groups from many amino acids in the liver. Okay, so we just showed that. So glutamate has collected amino acids from all over the place. Okay, in the liver. The amino groups have to be removed from glutamate in order to be excreted. Okay. that was happening in the cytosol. Okay, so all outside this gray area is considered the cytosol of our cell. Again, this is a mitochondria. Okay, so we have the outer membrane going around, and then we have this inner mitochondrial membrane. Okay, so inside the matrix.
And the cytosol would in liver cell or?
Yes, now we're yeah. So glutamate is going to travel from the cytosol, okay, to the mitochondria, and there undergo oxidative deaminization, okay. So glutamate gets the amino group removed. This is going to be catalyzed by L-glutamate dehydrogenase, okay. And that's what's being shown here on the next slide. So again, we were seeing this part, this is what we're looking at. So we just collected all these amino groups in the form of glutamate. That happened in the cytosol. Then glutamate is going to travel into the mitochondria where it's going to get deaminated by this glutamate dehydrogenase. Okay, that's the next step we're looking at is right here, which is shown again in this slide right here. This is oxidative deamination. Again, this is as it says in the title, this is catalyzed by glutamate dehydrogenase. Notice the name of the enzyme, right? Dehydrogenase. Notice it says it's an oxidative deamination. Okay, so we're oxidizing our substrate. Okay, so we're going from glutamate to alpha ketoglutarate. Okay, notice NAD plus is going to NADH, or NADP plus is going to NADPH. It's getting reduced depending on what enzyme. Okay. And then you're left with alpha ketoglutarate, and you're also inside the mitochondrial matrix now. Right? Remember, that's where citric acid cycle is also happening. Right? Citric acid cycle happens inside the matrix of the mitochondria. So we just moved that carbon skeleton in, right? We removed the amino group to go through excretion, and now we have that carbon skeleton left over that can feed into citric acid cycle. Okay. And it can be used for gluconeogenesis. Okay. And like we said, this ammonium gets removed, it's going to get processed into urea, that will be for excretion. And again, this is the start of ammonia excretion from our body. Okay. All right. Let's see. This is what I have on here for you guys. So where glutamate is transported from the cytosol of, I put hepatocytes on my thing, but liver cells, okay, into the mitochondria. What's happening? We have this oxidative deamination. Again, that glutamate, right, is getting deaminated. It's also getting oxidized. And we're left with alpha ketoglutarate. And then I circled that amino group and says, where does this go? Where does it go? Urea cycle. Okay, perfect. Okay, great. So again, we go back to this figure because you'll see, yeah, I'm sorry, Christian, what?
So I was just gonna ask, I got a little confused. because
Yes.
I understand that this is to deaminate glutamate to regenerate alpha ketoglutarate to do this cycle over and over, but I think it's called the alpha.
No, we're not in the urea cycle yet. We're not there yet. We haven't gotten there yet. I know. Seems like we have. Yeah, so remember this reaction that's happening. This one is happening in the cytosol. Okay, you have to remember that. Think of the. I feel like it's more bio, but think about the cell. We have the cytosol, which is just like all the liquid, right? And then we have the different organelles in the cell. And one of them that we talk a lot about in this class is the mitochondria. Okay. So this reaction was happening in the cytosol. Okay. Remember, there's no citric acid cycle happening in the cytosol. Okay. So we took an alpha ketoglutarate from the cytosol. It picked up the amino group from the amino acid and got transformed to glutamate. Okay. Glutamate then so that's, you'll see this figure in your book, we'll refer to it a lot. That's happening out here in the cytosol, outside of the mitochondria. Remember, the mitochondria has those two membranes that are really particular. The inner membrane is really particular about what gets in. Okay, there is a transporter that brings glutamate in. So we bring glutamate in, we remove the amino group. Now that will start to go through urea cycle, which again, we haven't looked at yet. Okay, but that's going to start to go through urea cycle. The leftover alpha ketoglutarate here, that could feed into citric acid cycle. But it's not connected to the alpha keto. Well, I mean, it is, but right, there's not like a direct, like we're not remaking this one, right? This is cytosolic alpha ketoglutarate. This is mitochondrial alpha ketoglutarate. Yeah, this is definitely one of those, like, it's hard to keep track of like what's happening. So again, we looked at how amino acids transfer amino groups to alpha ketoglutarate. That gets transformed to glutamate, and then glutamate gets transported into mitochondria. And we just looked at this oxidative deamination, removal of that amino group with that dehydrogenase. Okay, that was this. So again, all we've looked at so far is this piece and then into this piece. We haven't looked at this alanine, we haven't looked at this glutamine yet, and we haven't looked at this other parts, not yet. Questions? See why I made skeletal outline notes later, but and then stay with me here. Okay. All right. Okay, so then we're moving on to glutamine. Okay, so and in your book it says, in your worksheet it says where other tissues, not the liver, okay, maybe in the brain, okay. What are we doing? So in other tissues, right, if you have, again, like if you were breaking down your proteins because you're starving or you're untreated diabetes, and you had to transport that ammonia to the liver for excretion because it's toxic. Okay, so ammonia is toxic to us. Okay, so ammonia is toxic to animal tissues. Okay, but in those other tissues, when we start to break down amino acids, we start generating free ammonia. Okay. So what happens is that free ammonia, okay, can combine with glutamate. So that can combine with glutamate, and then that is going to form this glutamine. Okay. So notice here, okay, here's L-glutamate, okay? Again, there is the alpha carbon with the amino group and the carboxyl group and the hydrogen. Here's the R group, right? 2 methylenes and then this carboxy group, okay? You invest in ATP so that it can undergo this reaction with ammonium. Okay, and now we've added, right, this group here and formed glutamine. This happens, again, the enzyme that is carrying this out is glutamine synthetase. Again, take note of the extra syllable there. It's not synthase, it's synthetase. Again, that's an indication that ATP is required. You can see ATP required in the first step there. And notice that this excess ammonia becomes part of the, it's not part of the backbone, right? It's part of the R group. It's part of the functional group, okay? And so we can transport this buildup of ammonia through our blood with glutamine. So then glutamine will be transferred through the blood to the liver, where then it can undergo deamination, and then that excess nitrogen can be processed to urea. Okay. So again, this is in other tissues. So looking at this figure again, okay, that is this part. Okay. Glutamine. Again, notice everything is bringing excess nitrogen to the mitochondrial matrix of the liver for excretion through the urea cycle. That's what we're building up here too, okay? Any excess nitrogen, okay, is being transported to the mitochondrial matrix of the liver, right? In order to undergo excretion through the urea cycle. So what we just looked at was this, okay? Nitrogen from extrahepatic, meaning tissue that's not the liver. Tissue outside of the liver, okay? So from extrahepatic tissues, glutamine is bringing that the extra amino groups, okay? And then that can be transported in the mitochondrial matrix of the liver, okay? And again, amino group is removed once, and you're left with glutamate, and then another amino group is removed, and then from glutamate, you're left with alpha ketoglutamate. Okay. So then you have two, did you guys get those two boxes on that page? Glutamine synthetase. Okay, so glutamine is a non-toxic transporter of ammonia. Once in the liver, it goes to the mitochondria, and gets converted back to glutamate, right? All ammonia ends up at the liver. Well, not true. But it goes through the urea cycle if it's not used in biosynthesis. Okay, it's not used in biosynthesis. Okay. All right, I say the best for last. Okay, comments. The next one, this one I love this one. This one shows up on the test too, because I like it. Okay. So just be aware. Okay. Because I think it's just really cool. So notice on this figure, we got another source of nitrogen coming from alanine from the muscle. Okay, coming from alanine. Okay, so if you're working out muscles, okay, and they're operating anaerobically, meaning with very little oxygen, without oxygen, okay, and they're relying on glycolysis for energy, okay. Remember, after you go through glycolysis, you get pyruvate. Okay, we know that we have a pyruvate. We know that if pyruvate undergoes fermentation to help regenerate NAD+ for glycolysis, okay, then pyruvate will form lactic acid. Okay, this starts to build up. Okay, but that pyruvate can be converted to alanine. Okay, I don't have the structures here. Okay. So here's your muscles, right? You're burning up your glucose, going through glycolysis, you're forming that pyruvate, okay? Normally that pyruvate might undergo some fermentation, okay? But that pyruvate can also be converted to alanine. Okay, so pyruvate is the alpha keto acid of alanine. Okay, just like we saw glutamate was the amino acid and alpha ketoglutarate was the alpha keto acid. Okay, that's the same relationship between pyruvate and alanine. Alanine's the amino acid, pyruvate's the alpha keto acid. So you just remove the amino group from alanine and you get pyruvate. Or in this case, again, if you're breaking down your muscle protein into amino acids, you get that extra amino group to glutamate. Glutamate transfers the amino group to pyruvate and you form alanine. Notice the name of the enzyme, alanine aminotransferase. Okay, transfers the amino group to alanine. Alanine can safely transport through our blood. Okay. And once alanine gets to the liver, it can go through that removal of the amino group, again, with the same enzyme, alanine aminotransferase. But this time you're taking the amino group from alanine, transferring it to alpha ketoglutarate. You get glutamate, we've seen that a lot. But remember, from alanine, when we remove the amino group, we get pyruvate. And then look at pyruvate is there in the liver, ready to undergo gluconeogenesis. So then you can make glucose from that pyruvate, and then the glucose can be transported back in the blood, back to the muscle that needs it as a fuel because it's vigorously contracting, okay? And it can go through this cycle again, okay? It's like transporting amino groups with alanine in the blood is like 2 birds with 1 stone. Like you're taking the amino group to the liver to get excreted, and you have the carbon skeleton, which is pyruvate there, to undergo gluconeogenesis, to provide more glucose for the body. Isn't that really cool? So this is the glucose alanine cycle, okay? Let's see, amino groups donate to pyruvate to form alanine. Glutamate is converted to glutamine and this and transported to further. Oh. What glycolytic product and muscles produce? That'd be pyruvate. Okay, and then you got the back of the sheet. Pyruvate gets converted to alanine. Passes to the blood to the liver. Transporting ammonia as alanine gets the pyruvate and the amino group to the liver. What can the pyruvate do once it gets to the liver? Gluconeogenesis. Again, ammonia is a product of amino acid breakdown. It gets excreted by the urea cycle. Yes. Is that a lot?
Yes.
Okay.
I didn't get the glutamated converted glutamine and transported to
The liver.
thank you.
Yeah. Oh, pyruvate. So notice, let's look at the structures real quick. So notice between alpha ketoglutarate and glutamate, right? Again, this is the alpha keto acid, this is the amino acid, okay? So what, like it's the same. Ah, here's the difference, right? Here, it's a carbonyl, and here, we've added the amino group and a hydrogen, okay? Again, this is the same. So that's the difference. So on that last page, you have the structure of pyruvate, but you don't have the structure of alanine. That's what you're supposed to fill into that box. But you can do that if you have the carbon skeleton, okay? Because again, I told you pyruvate is the alpha keto acid of alanine. So again, you see pyruvate there, it's you have the carboxyl group, a carbonyl, and the methyl group. If you change the carbonyl, right? So instead of that carbon being double bonded to oxygen, it should have a single bond to hydrogen and then a single bond to an amino group. You guys see what I'm saying on this last part up here? You have the structure of pyruvate. Right? Take that carbonyl part of pyruvate. Right? And aminate it, make it look like this. That's the relationship between these alpha keto acids and these amino acids. Okay, we go from a ketone to an amino group. Okay. Ooh, man, yeah, that part is right before testing. That is usually how it works. Again, the whole point of today's lecture was how do these different, where are these different sources of amino groups? How do they get into the liver? Again, either for excretion through the urea cycle, which we'll get into next week, okay, or for biosynthesis. That's all it was. That's all. I know it was a lot, but how do we get these amino groups to the liver? Whether we've taken them into our diet, whether we're breaking down muscle protein, okay, or we're just some other tissue that has some excess ammonia. How do we get those amino groups to the liver? Because again, they're toxic, we need to excrete them, unless we're going to use them in some kind of making or something. Right, questions for me? Okay, yeah.
How many questions was the end of the same?
It's there'll be no Scantron, you can just write on the test. It's similar, like. Yeah. Let me stop my recording. All right, you guys.